back to your next installment of APES review for the AP Enviro exam. This is Andy. Today I'm going to talk to you about lakes, lake experiments, and kind of related to water quality. A little bit of my background: in college, I was fortunate to work at a limnology research center in northern Wisconsin. Yes, I do have to give you a definition of lakes. It's a non-moving body of fresh water. A moving body of fresh water would obviously be considered a river. A lentic ecosystem means that you're dealing with a lake. Limnology started with the help of this professor from UW-Madison in the first part of the 20th century. Art Hassler, actually maybe even mid-part. A lot of people think he might have even just done it all just to get the university to pay for his fishing trips in northern Wisconsin. And initially he just used the same sampling equipment as saltwater, thinking that lakes would just be like the ocean, only without salt and without tides. He ended up creating a whole new science. As we talk about lake studies, it's a good time to also talk about observation and science. You can have two ways of doing science observations: qualitative, which is things like opinions, observations, images. You could say a lake is muckier than another lake. Quantitative is numbers. We often do this mistake in science where we think everything has to be numbers and everything has to be a graph. In good science, you actually use both. And sometimes you have to recognize when one form of data is better than a different. When we talk about water sampling, one of the things that they do in limnology is this is the holy trinity. Turbidity is how mucky or murky the water is. Temperature is obviously temperature. Just, temperature is the measure of movement of particles. Dissolved oxygen is how much oxygen is dissolved in the water. That's usually measured with things like Winkler titrations, snap test, or sensors. The Winkler titration is the best. If you were in my class, I would be showing you how to do a Winkler titration. I used to do it on research boats, and the term for completing one is winkling. So those are the major ones. There's other things you can measure as well, but those are the big ticket studies for water quality. So here's turbidity. The more stuff that is suspended, like algae in the water, tells you a lot about turbidity. When we talk about eutrophication later, weird thing is one of the best ways to tell if a lake is eutrophic is before you can even measure the phosphates, nitrates, or dissolved oxygen, you can just look at the water and tell by its color. Here's an example of cultural eutrophication. Lake Tahoe. Its water clarity is how far you could lower a Secchi disk. The way you actually do this is you lower a black and white disk until you don't see it. There are electronic sensors that you probably have in your classrooms. They're not as accurate as a Secchi disk. Basically, it's a frisbee of science. Lake Tahoe in the 1960s had water clarity of about 30 feet, which means if there was or 30 meters, which means you would not see the lake monster till it was about 100 feet away. 100 feet or 30 meters. More recently, because of golf courses and shoreline development and leaking water treatment, including septic tanks, it's down to about half of that, which means now the invisible lake monster, or the lake monster, will get quite a bit closer to you before you see it. There are no lake monsters in Lake Tahoe. So there's no diagram. This is actually how a Secchi disk looks. And when I worked on research boats on the ocean, we were often lowering these things down over 100 feet to 200 feet, especially in tropical waters. In Lake Tahoe, 30 meters was its original one. A lot of smaller lakes in the Great Lakes region, their natural turbidity or Secchi depth is about 6 to 10 feet. Temperature, we use in lakes to measure the entire temperature column, starting at the surface and going down a meter or a few feet, and recording the temperature again and again. The surface layer is called the epilimnion, meaning epi meaning surface. The deep layer is called the hypolimnion, which is the deeper part. This time of year, if you're wanting to go in the water, you might be noticing this. The first part of the water that warms up is obviously the surface. Warm liquids are less dense, so the warmer water accumulates on the surface. That's why this time of year, when the water starts opening up and the ice melts, if you stand ankle deep, the water might feel warm. If you go knee deep, it's freezing. The reason why is as you go deeper, you're hitting colder water. This is also why people think the middle of the lake is colder. It's actually the bottom of the lake is colder, but when you're in the middle, your feet are often touching that colder bottom layer, cooling down your entire body. Dissolved oxygen is the oxygen dissolved in the water. There's a lot of things that affect dissolved oxygen. If you like things like, say, salmon and trout, they need really high dissolved oxygen. These are the major influences. Lake size. Bigger lakes tend to have higher dissolved oxygen because there's more area for the wind to act on. Light intensity, because first of all, light does cause photosynthesis, causing dissolved oxygen to go up, but light also heats up the water, and warmer water dissolves less gas. This is why restaurants freeze hot water in their ice makers. Not only does it make clear gas, but by boiling the water and then freezing it, it uses less energy than trying to freeze cold water because the dissolved gases slow down the freezing process. And wind. The more wind that blows on the water, the more dissolved oxygen you get. And nutrients do a weird thing. As we add nutrients, we add to cultural eutrophication, which ultimately brings down the dissolved oxygen. And today is the magical day where we get to make the word fetch happen. So to help with that, I'm going to show you some pictures of lakes. Here are a few lakes in northern Wisconsin where I grew up. This is Star Lake. My buddy Will has a resort right here, and he's not willing to admit that the resort used to be a brothel. This lake right here is called Plum Lake. These two lakes are about the same size. Plum Lake has a higher dissolved oxygen because the lake starts here and ends here. The longest line that wind can act on is called fetch. The more fetch you have, the more wind action you have, the bigger the waves are. You can also see this on this lake. The prevailing wind usually blows from west to east. As the wind blows, by the time you get here, it has giant 3 to 4 foot waves. On this end, the water stays pretty calm, so it's good for water skiing and canoeing. Right here, by the way, is a summer camp, and one of the coolest things to see in the summer is they're protected from the wind, and when they do their sailing days, often they don't realize how strong the wind is until they get out here. The reason why it's so windy here is because this lake is so fetch. As a result, it has higher dissolved oxygen. Here, by the way, is Art Hassler and one of his assistants or fellow professors doing some research. Funny thing, the reason the way they took this picture is this was all staged. No one would be that dressed up doing research even back then. And obviously the photographer was standing on a pier or a dock. Here's one of their old research labs. Over the past 70 years of freshwater research, limnology, we've changed a lot of scale, which is a cool way to understand science as a process. One of the things you have to do in your APES FRQ, if there's a design and experiment, is they're looking for ways to increase your data set, doing things like replicates or multiples. Ways that they've changed the scale in lake research is they've started comparing hundreds of lakes for the same variable. There's online databases that you can look up, and if you want, you can start researching data on almost any lake you can find on a map. They also changed the time scale, which I'll show you. In the past 20 years, they've added a new thing where they're increasing frequency. These are all ways to expand your data set. Here's an example with number of lakes. So here's Star Lake, Plum Lake, and the lake I grew up on. Let me move this. Big St. Germain. These lakes have 3 different amounts of clarity. Star Lake, as you can tell, is incredibly clear for this region, 20 feet. Plum Lake is only 12 feet. Big St. Germain is 8 feet. It took a while to figure out why these lakes have different clarities because they're all about the same size. Ultimately, what it is Star Lake is about 6 inches higher in elevation than Plum Lake, which is about a foot higher in elevation than Big St., which means all of the water from this whole picture flows into Big St. and can carry more nutrients. So here is, by the way, this is awesome. This is a solar-powered lake buoy that measures water quality every foot of depth going from the very surface to the very bottom, and it measures every hour. It's this program called GLEON. Another thing we'll talk about is long-term ecological research. This is expanding our data set so that we can actually measure things not for days, not for weeks, but for decades. In ecology, scale is a lot slower. Long-term ecological research are research programs, and this is the Northern Lakes one in northern Wisconsin. The Northern Temperate Lakes Long-Term Ecological Research Center is its full term. They are collecting data all summer and all winter with just the goal of stockpiling it in case there ever is a researcher that wants to use it. I got to spend a whole summer just collecting behavior notes on smallmouth bass. The next year, I measured the amount of coarse woody debris or driftwood in lakes. This data ended up getting used about 2 decades later. Here's how changing scale can show you stuff. Here's dissolved oxygen over a few hours. As you notice, there isn't much trend. This right here is a windstorm. If you look at that same lake, you'll see the dissolved oxygen over the scale of a few weeks goes down. The reason why is as the water warms up in the summer, dissolved oxygen goes down. And another cool thing is there is a bigger windstorm. Here's another trend. There, you don't really see too much happening with dissolved oxygen. Right here is an awesome trend where you see, once again, dissolved oxygen goes down in this lake every summer. This is Trout Lake. By the way, this was a whole series of tornadoes. I remember because it crushed my car. No researchers were hurt. This data was collected by a buoy on the middle of the lake. One other thing about using these buoys is we're no longer putting researchers in danger because this buoy can collect in a split second the data that would take a group of biologists hours to collect otherwise. So here's what Gleon does, and it's an awesome thing for collecting lots of information. It's free data for everyone as well. Now, let's talk about lake layers. We talked about the warm surface layer called epilimnion. The deeper water is called the hypolimnion. And this is obviously the surface. The area where the temperature changes is called a thermocline. And here's the weird thing. The hypolimnion on a lot of lakes is around 34 degrees Fahrenheit. The epilimnion on lakes can reach a comfortable swimming temperature during the summer. This boundary could be just a few inches thick. And the change in water density causes light to refract. And sometimes when you're diving down to the thermocline, if you're wearing goggles or a mask, you can actually see this layer shimmering. All summer, the thermocline goes deeper. And here's the strange thing. This surface layer never mixes with this deep layer. There are such different densities. So when wind blows, the epilimnion gets exchanged with the surface, but the hypolimnion gets nothing. There's only a few times of the year where this can collect oxygen from the air. The littoral zone is where sunlight reaches the bottom. If you were like me when I was a kid, I did not like swimming in weeds, so I would frantically swim past this point until I got to deeper water because I was convinced the weeds were going to wrap around my legs and strangle me. They won't do that. So the other term for the thermocline is called the metalimnion. So the epilimnion in the summer is warm. The hypolimnion is cold. And if you've ever gone in a lake and dove down, you've often encountered the frigid layer that is the hypolimnion. So another way we study lakes is how they mix. Lakes can mix a few ways. Amictic means they never mix. Monomictic means they only mix once. Dimictic means they mix twice. Polymictic means they mix many times. This is an important thing to know because the only time a lake mixes is when the entire water column from the surface to the bottom is the exact same temperature. Once a thermocline forms, they stop mixing. Amictic, this is a volcanic lake in Africa called Lake Nyos. It's freakishly deep and it doesn't mix. One thing is anything that sinks there decomposes and produces carbon dioxide. But as you go deeper in the water, there's pressure, so that carbon dioxide remains dissolved. Unless there is a mild earthquake, which happened about 10 years ago, and the entire lake fizzed out carbon dioxide and suffocated the surrounding community. To reduce that danger, lakes like this are often get aerated now to reduce that chance of loss of life. Monomictic are subtropical lakes, which would be places like Florida. They mix once during the winter, but the lake doesn't freeze. Polymictic are tropical lakes, which are mixing usually after rainfalls. And because they're tropical, there is no winter cooling, so these have frequent mixing. Dimictic mixes twice. So right around now, if you look at a lot of these lakes, as the ice goes out, they're doing their spring mix because the entire water column is about 34 degrees. If you look carefully at the water, you'll actually see little sparkles from these microscopic algae called diatoms that make the lake shimmer and slightly change color for about 2 days. Then the surface layer starts building up and you get your summer stratification. It mixes again in the fall just before the lake freezes. The freezing is considered another form of stratification. The only difference is when the lake freezes, the cold layer is now on the top and it's called ice. Now, pollution is any change to the water. Don't just say pollution on the AP exam because you'll sound like a vapid beauty queen. No offense to beauty queens that are smart. Thermal pollution, oil, these are the major pollutions that we often talk about on the exam. Thermal pollution is changing the temperature. Usually that's making it hotter. Oil and gasoline can cause problems in water quality. Acid rain, which I'll show you some experience about that. Light pollution affects a lot of nocturnal animals and that would otherwise would benefit from darker things, which includes just, birds of prey like owls around the lake. As well as invasive species, which is any species that was introduced or is out competing others. So before we get into that pollution, let's go over lake zones. Now, riparian zone is the shoreline near the lake. According to most lake scientists, this goes back about 200 feet because that's how tall a tree could grow that could fall into the water. Photic zone is the area that has light. Where the photic zone overlaps the bottom is the littoral zone. This is where most of the smaller invertebrates and life is found in the lake. The benthic is the actual bottom. Pelagic is the open water area, which is good for water skiing. So there's a prettier picture of it. And photic and epilimnion don't necessarily overlap. So let's go back to the pollution. Thermal pollution. This decreases dissolved oxygen as you warm water temperature. This can come from things like power plants or just runoff coming across parking lots and streets in neighborhoods. Even just things as simple as removing the trees from a shoreline can cause the water to increase in temperature. The other thing is it increases respiration rates in animals and increases their metabolic rates. Even weirder, in Lake Mendota in the 1980s, they found several piranha that had overwintered because they had found the hot water pipe outlet for a water treatment plant. The worst part was they don't know if they reproduced or not. So for about a decade, people were nervously wondering if piranhas were going to get established in Lake Mendota. They did not get established in Lake Mendota. Oil and gasoline, the biggest source from this is usually not oil companies having spills. The biggest source is leaky engines and parking lot runoff. Now, acid rain. On the APES exam, as you know, SOx and NOx combine with water to form sulfuric acid and nitric acid. Sulfuric acid or SOx comes primarily from burning coal and diesel. The NOx actually comes from hot car engines. And rather than the cliffhanger question, this area right here is the area where the lakes have been most affected by acid rain. All right, so acid rain became a problem in lake science in about the 1970s. They noticed when lakes went acidic, the entire fish population vanished rather quickly. It wasn't a major change in pH. Normally, freshwater is a pH of around 6. Acidic lakes were getting to be a pH of around 5. That's not enough to dissolve fish, grandma, or anything. So my professor in the 1980s came up with a really awesome experiment with the help of one of his grad students. They decided to make a lake acidic just to see what happened. So they worked with this lake called Little Rock Lake. It's right off of Highway 51, just north of the town of Minocqua. They made a plastic shower curtain right there and divided the lake in half. Then they made one half incredibly acidic over the course of 5 years. It was done by adding sulfuric acid in small amounts through modified outboard motors that had a funnel on top, and the propeller would mix the sulfuric acid in the lake. They created a schedule based on the volume of the lake to slowly add sulfuric acid once every few days in small amounts to make this lake go acidic within 5 years. This half, they did nothing. So they had a control and an experimental treatment. The reason why they did this slow addition of acid process is it would have been faster to bring in truckloads of sulfuric acid, but that is not how a lake becomes acidic. After 5 years, the pH hadn't changed at all. The professor's involvement with the experiment ended up just adding more and more acid until they finally figured out why the fish died when the lake became acidic. The lake was closed off to the public. There was a little gate that prevented any access of the lake to anyone but researchers, causing a lot of conspiracy theories in this community. And by the way, this is a fish from an acidic lake. This is a fish from a non-acidic lake. Ultimately, what they found out was it was a trophic cascade. If you've watched any of the videos about trophic cascades, when the lake became acidic, the zooplankton, the base of the food chain just above phytoplankton, changed. The new zooplankton that could handle the acidic environment was a different species that nothing else liked to eat, causing the food chain to collapse. The reason why this was not found on a smaller scale is when they did this with aquarium studies, the fish were fed, so they didn't realize why the fish were dying off. They ended up doing this experiment for decades. Once they figured this part out, they also realized some lakes in the Great Lakes region, like Wisconsin, have bacteria that neutralize acid to a certain amount, and that's called biological buffering. They stopped adding acid, and then they discovered over the course of a few years, the pH of the lake returned to normal, and the food chain did recover. So another way to differentiate lakes, just so you know, is nutrient loading. Oligotrophic are low nutrients, clear water. These are the ones that are great for swimming. These are the good ones that are good for looking for wildlife. Oligotrophic and eutrophic both naturally exist. When humans add nutrients, that's called cultural eutrophication. And I'll show you some examples of that. As you add nutrients, dissolved oxygen goes down, turbidity goes down. The clarity goes down, turbidity goes up. You get less big fish. You have higher temperatures. Not so good for swimming, sometimes good for wildlife, but if it goes too far, an oligotrophic lake can cause problems. Here's an eutrophic lake. Here's an oligotrophic lake. It's really clear. Just so you know, the blue is actually from it reflecting the sky. If you look at it from above, it's clear-ish. Here's eutrophic. This is the place where if you step, your feet are going to sink in muck. These are great for wildlife, especially birds and amphibians. You often see this diagram of lakes slowly filling up and becoming glades and ponds in textbooks. This happens on the scale of tens of thousands of years. We tend to have ice ages that prevent this from happening. These diagrams ended up in textbooks, and they often taught people, oh, lakes just naturally get green and scummy over time. That actually doesn't usually happen unless humans screw up the system. Hyper-oligotrophic, just so you know, are really clear lakes like Lake Tahoe. Hyper-eutrophic lakes are incredibly murky ones that will, just smell like sulfur and farts because of anaerobic bacteria. Mesotrophic are somewhere in the middle. And this is cultural eutrophication, is when humans add nutrients. We already talked about Lake Tahoe. The key thing on the APES exam is do not just say eutrophication. You say things like dissolved oxygen goes down, fish die off, the water gets murky, increased growth of algae. If you say a few of those terms to describe eutrophication, you get the point. If you just say eutrophication, they won't give you the point. And in the case of Lake Tahoe, you can see that it became eutrophic rather quickly with just human development on the shoreline. The cause for this is plants need three major nutrients: nitrogen, phosphorus, and potassium. In lakes and oceans, when we talk about eutrophication, it's usually phytoplankton, algae, which is now considered a protist. So start just saying protist. One weird thing is in oceans, nitrates cause algae blooms. We already knew this, so we assumed it must be nitrates causing it. A weird thing is if you test seawater for nitrates, it's usually 0 because it's the limiting factor and it tends to get absorbed by plant growth quickly. In the case of freshwater, you can detect nitrate levels. Usually when you detect freshwater for phosphates, you don't see any phosphates. So the measure of phosphates is algae blooms. What we now know is phosphates are actually the limiting nutrient in freshwater, and I'll show you the experiment that taught us this. One way to remember it is just think phosphates are fresh. The Canadian government, noticing lakes were going eutrophic in the 60s and 70s, did some really cool experiments. They found a pair of lakes. They added phosphates to one and nitrates to the other. The phosphate industries included laundry detergent companies. Phosphates are also found in car wash soap and dishwashing detergent. And when they added phosphates to one lake, that lake became eutrophic. The producers of phosphates said this wasn't enough evidence, and they had a good point. They said, you said you were doing your experiment because lakes were going eutrophic. How do we know that lake was not just about to go eutrophic? Yes, companies and adults are just as bad as all of you are sometimes. So the Canadian government found another lake, and they divided it with a plastic shower curtain. This is the half that got phosphates. This is a really amazing photo and a great moment in history for aquatic science. And by the way, this plastic divider, if that sounds like the experiment that was the acid rain experiment about 2 decades later in northern Wisconsin, the reason why is the grad student's father was a professor in Canada, and he just replicated this when he did the acid study in Wisconsin. So currently, laundry detergent in North America no longer contains phosphates. Phosphates are still found in car soap and dishwashing detergent. Your dishwasher doesn't really produce too many phosphates, and modern washing machines prefer an enzyme-based soap. So you're now seeing most dishwashing detergents, the companies are voluntarily removing phosphates from it. Because it's found in car soap, this is why you should never have a car wash for the environment. So phosphates aren't entering the water as quickly. Septic tanks are getting replaced in a lot of the region, further improving water quality. UW-Madison ended up finding another way to clean up lakes that had gone eutrophic. Lake Mendota is incredibly green and scummy. In the 80s, they discovered if they added more large fish predators, so things like muskie and pike, that would cause a trophic cascade, ultimately resulting in lots of zooplankton eating down all of the algae. It was a great experiment, but then everybody found out the lake was loaded with trophy fish, and recreational fishermen fished it out almost overnight. There's Lake Mendota, by the way. So the way we usually test for phosphates is through things. The best thing to do is actually doing something as simple as a Secchi depth. Another good approximation of phosphate levels in fresh water is just dissolved oxygen. Otherwise, do Secchi depth or turbidity. So here are other sources of lake nutrients or culture eutrophication: soil erosion, fertilizers, and the big culprit tends to be residential and golf courses. You can't pick on the farmers. Farmers tend to not overuse the fertilizers the way homeowners and golf courses do, as well as old septic tanks. What you can do to help improve water quality in lakes and as well as rivers is try to make sure you're using a low phosphate dishwashing detergent. Wash your car at a car wash, not on the street or in your driveway. That when you wash it in a car wash, that water actually gets captured and goes to water treatment. Also put in things like rain gardens or other things to absorb runoff to help keep it from going directly into the lake. Now, here's one last tangent about fish, and this is a weird one. In the 1970s, a few reservations in Wisconsin sued the federal government for their treaty rights that allowed them to spearfish. And it led to some of the most racist, embarrassing things happening. And it was where a bunch of racists and vacationers all collided over, of all things, fish and lakes. These are some pictures about protesters. I remember this growing up in the 80s when this really hit the spotlight, and it was embarrassing. There were school administrators in this district that were caught throwing rocks at people who were spearfishing, even though they were allowed this through treaty rights. And the people spearing were actually students and parents in the community. It was a horrible moment. The reason why a lot of these people jumped into racism is they were convinced that spearfishing was going to take away all of the fish in this region. The weird thing is spearfishing only accounts for about 5% of the fish harvest in the state of Wisconsin. This experiment that resulted didn't happen for about 25 years. It was a coarse woody debris experiment. It happened in Vilas County, and they took a pair of lakes. What they had already noticed is homeowners, whenever they buy lakefront property, they want to clean up the shoreline, and they take all of the logs out of the water. Sometimes they use them as ugly yard decorations. Other times they just like the lake to be tidy. This first started getting data studied in the 1990s. I was part of it, and they quickly realized whenever there were more homeowners on a lake, they found less coarse woody debris in the littoral zone. The year I actually helped the study, we actually put survey markers in every large piece of coarse woody debris, and then each year we had to go and find them. The year I went, a bunch of them were already missing, but we found them with the survey marks in people's rock gardens. So what Trout Lake Station did in northern Wisconsin is they took another pair of lakes on a research area. There are no people allowed there other than for research. In one, they got a bunch of people to remove every piece of coarse woody debris from the lake. In the other one, they had a timber company haul out all of the coarse woody debris from a timber operation and put it on the shoreline of another lake. And then they monitored the fish populations. Initially, both lakes had identical fish populations. Within 3 years, the one where they loaded it with coarse woody debris, the fish population skyrocketed because it provided shelter for the young fish. The one with no coarse woody debris, the fish population plummeted because there was no longer replacement of or new fish that were surviving. The reason why this experiment was kind of groundbreaking is it kind of changed how people interacted with shorelines. The other thing they realized is the fish collapse in the 1970s in northern Wisconsin was not caused by spearing. It was not caused by recreational fishing, overharvesting. It was actually caused by all the homeowners, the very ones that were screaming racist things at the Native Americans. The homeowners in the 70s started removing all the coarse woody debris because there was a housing boom in this region. So one other challenge that we have to talk about with water pollution is each test is expensive, and you need a separate test for each chemical. But there's another way you can test water quality, and if you're careful, this is something you could do while you're under isolation right now. You look for macroinvertebrates. There's programs online that tell you them. The key is looking for the big ticket super water quality heroes of water quality. When you see things like mayflies and stoneflies and dragonflies, that means you have really good water quality. So that was it for water quality. I hope that was a good review. Water quality should still be, relevant on the AP exam.